DNA is the building block of life. DNA encodes elegant structures that are used as templates for our cells, almost like words in a dictionary. Yet, despite our attempts to read this dictionary, we still don't know the exact effect of modifying specific genes of our DNA. CRISPR can help us with our problem. CRISPR is a nuclease system that provides a precise and convenient tool to manipulate the DNA of our cells. Specifically, CRISPR allows us to cut and paste DNA at specific locations in the genome, almost like cutting and pasting into a Word document. This tool has many applications for the future.
CRISPR has a wide range of applications in research. It allows us to directly modify a gene to study its effects in cells. In the Agarwal lab, we are studying the TERK gene, which encodes the RNA component of telomerase. Telomeres located at the ends of chromosomes shorten with each successive cycle of DNA replication during cell divisions. TERK contains a template used for lengthening telomeres. Mutations in the TERK gene can lead to many genetic disorders such as dyskeratosis congenita, a rare genetic disorder characterized by rapid shortening of telomeres. To better study the effects of specific mutations, we use HEK293 T cells, a well-established human embryonic kidney cell line that is easy to culture, to manipulate the endogenous TERK gene. Previously in the lab, a cell line was created with a TERK deletion mutation that is characteristic of dyskeratosis congenita. These cells deteriorate and die with increasing passage number. Using the cell line and CRISPR technology, we can do a number of things to study the TERK mutation. We can insert template DNA with CRISPR to restore portions of the deleted region or the entire deleted region itself. We will then be able to see how the cells are affected in culture and whether the manipulation was sufficient to rescue the cells and restore TERK function.
Another way CRISPR is used in research is to detect when a gene is expressed in cells. In the Hyman lab, we are trying to learn more about psychiatric disorders such as schizophrenia. Despite its prevalence, the etiology of schizophrenia is poorly understood, partly because of our inability to readily study neurons. If we can readily study neurons, specifically cortical pyramidal projection neurons that are found in the brain, we might be able to learn more. We are refining a procedure to differentiate induced pluripotent stem cells, or iPSCs, into cortical pyramidal projection neurons. Our main problem is that we need these specific neurons to study. This isn't easy. We aren't guaranteed to get the correct neuron from differentiating iPSCs. Through a lot of work, protocols have been developed that are meant to maximize the production of the correct neurons. But how do we know if our protocols work? What we can do is, using CRISPR, put an indicator into our iPSCs. If we put the indicator next to a gene that only our desired neurons express, we can detect when it is expressed, letting us know if our protocol succeeded. This will let us refine our protocols, ultimately giving us a readily available number of cortical pyramidal projection neurons we need.
CRISPR genome modification has shown great promise. It works well on a small scale when applied to cell cultures, and it potentially can be scaled up to uncover therapies for dyskeratosis congenita and schizophrenia.